Alright, there's a lot to this section, so we're going to see how far we can make it in these 30 minutes. So we're basically dealing with genetics. Here's an interesting kind of introduction. The people of Tibet live and work in altitudes above 13,000 feet, where the amount of oxygen that reaches the blood is 40% less than at sea level, which is basically where we are. So that makes the Tibetan people so able to tolerate these harsh surroundings. Over the last several thousand years, the Tibetan population has accumulated several dozen genetic mutations that affect their circulatory system and their respiratory system. So these are the Tibetan people. Alright, so how do we get there? We've got to actually figure out a little bit of Mendel's laws and some variations. We won't do a ton with the variations today, but we'll start with Mendel's laws for sure. This is going to be the biggest part. Our essential question today, I'll get it written down here, will be, what are Mendel's laws of inheritance? So that's one. And two, how can we use crosses to determine genetic probability? Probability or possibility, either is a perfectly good word to kind of put there. Okay, so just some ancient roots here is that the Greek physician Hippocrates explained inheritance by proposing that particles called pangenes travel from each part of an organism's body to the egg and the sperm. And then the characteristics acquired during the parent's lifetime can be transferred to the offspring. So he had this first idea that you could kind of transfer things to the offspring. Hippocrates' idea is incorrect in several aspects. So the reproductive cells are not composed of particles from the somatic body cells. And changes in somatic cells do not influence the egg or the sperm. So it's a little bit off there, but at least the idea that started was the transfer of offspring. So there's old school Hippocrates. So the idea that hereditary materials mix in forming offspring was called a blending hypothesis. It was suggested in about the 19th century by scientists studying plants, but then later rejected because it didn't explain how traits that disappear in one generation could reappear in later generations. So we've all heard that when things like skip a generation, blending can't really explain that for us. All right, so some key terms. Heredity is the transmission of traits from one generation to the next. So it's just kind of transmitting, it's changing, it's moving traits from one generation to the next. Genetics is our scientific study of heredity. All right? So Gregor Mendel, this is our main kind of dude in action here. We're talking about Mendel's laws and things like that. We're talking about this man. So he technically began the field of genetics in about the 1860s. He deduced the principles of genetics by breeding garden peas, which he got super lucky with having chosen peas. If he had chosen just about anything else, I don't know that we would have Mendel's genetics. Okay? But he actually relied upon a background in math, physics, and chemistry in order to figure that out. So this is Gregor Mendel. He was an Austrian monk, by the way, which is why he's dressed so sharply. All right, so in 1866, Mendel correctly argued that parents pass on their offspring these discrete heritable factors. And he stressed that heritable factors, today that we call genes, retain their individuality generation after generation. So a heritable feature that varies among individuals, such as like flower color, is called a character. Each variant for that character, such as purple or white, is a trait. Okay, so we'll look at some of those examples, but the feature is the character and the variant is the trait. So if that's still a little wonky to you, we'll get to a picture here in just a second. So perhaps the most important advantage of pea plants as an experimental model was that Mendel could strictly control matings. Yeah, that's the thing with you could really control the mating. Plants are a little different than animals in that simply just it's easier to control. So the petals of the pea flower almost completely enclose the reproductive organs, the stamen and carpel, which we'll talk about later. So consequently, the pea plants were able to pea plants are usually able to self-fertilize in nature. So in nature, they do self-fertilize, which you can kind of use because I won't say even plants don't really self-fertilize in nature. So if you see, this is kind of where the eggs are, and this is where sort of the pollen is, or the sperm containing parts there. They're close together and sort of enclosed. So self-pollination is a thing that happens. Okay. So when Mendel wanted to cross-fertilize plants, he prevented self-fertilization by cutting off immature stamens of the plants before they produced pollen. And then he dusted some carpel on the pollen, carpel with pollen, to cross-fertilize. So a lot of background here, but the whole point is that he can control between cross-fertilization and self-fertilization. So he could control this. That's the basic premise here. He could control it. We haven't gotten into plants yet. That's next semester. But that's the whole point. He can control it. So after pollinating, which is sort of the plant equivalent to fertilization, the carpel developed into a pod containing some seeds or peas that he was later able to plant and figure stuff out from. So that's your background. Is what he could do is he could cut off stamens, he could dust pollen on a different thing. So he really, really could control that with a paintbrush and scissors. The magic of plants is that you could deal with the paintbrush and scissors and fix that whole thing. Okay, so true breeding varieties, and this part is important, is that there's true breeding varieties that resulted from self-fertilization and produced offspring that are all identical to the parent. Okay, if an offspring of two different varieties, we call those hybrids, and we'll see that term come up a lot. So the cross-fertilization is a hybridization. So whenever he crosses one plant with another, there's your plant, and he crosses one with another plant, there you go. He's able to sort of give the pollen to each of them and trade them. Then we call that cross-fertilization or hybridization. It's my terrible drawing because this marker doesn't work. Okay. So true breeding parental plants are the P generation. P for parental. The parentals in our case here are always true breeding. They're always identical. They're always just the same variety. So they're true breeding. Hybrid offspring are F1. F is what stands for filial. It's if I can even spell it right, filial. So filial is just a term for sons and daughters. Yeah, I can't write it all out there. Let's try daughters. Just catch it there. All right, so it says the first sons and daughters. So basically all this is the first offspring. So filial just means sons and daughters. It's the first offspring. Then, with these hybrid, is that Mendel was able to cross them with each other. They kind of crossed all those plants. You cross them again, and you get an F2, or the second filial, or the second set of sons and daughters. So that's all we're going to have. Parental, first set of sons and daughters, second set of sons and daughters. That's it. Okay, back to characters and traits, which are going to be important for this, is that here's some examples of characters. So like flower color, flower position, seed color, seed shape. Okay. Different traits are some different types. So for flower color, we might have purple and we might have white. Flower position, we might have axial, so on the side, and terminal, which is up top. And the dominant and recessive we'll get into. So those are coming. All right, just know that there's some different variations. All right, so a cross between two individuals differing in one single character, So from that last list that we looked at, like maybe we do flower color is the one character we look at. No other character is involved. I almost wrote character color. No other thing is involved, but just one particular character. We call that a mono or one. Jeez, I can't write. One hybrid is kind of a cross-fertilized cross. So one single hybrid cross that we're looking for. Alright, so Mendel performed a monohybrid cross between a plant with purple flowers and a plant with white flowers. Okay, so these were true breeding purple and white. In the F1 generation, they all had purple. When he crossed the F1 plants with each other, what he found was 3/4 were purple and 1/4 were white. So the white jumped a generation there. Alright, and that's where we kind of sit at. That's a good place right at 11 minutes, so it's a good time to take a break. Chunk your notes, look at them, write some questions, come back. Okay, so here's what it looks like. P again is the parental generation. These are true breeding. So all the purple flowers here came from purple flower backgrounds. White flowers all came from white flower backgrounds. Okay. When he crossed the two, all of them ended up being these like purple. We didn't find any white anywhere. It was just purple. Then when he took these guys, these first sons and daughters, and crossed them together, because you can do that in plants, they're okay with it. Okay, so when they crossed those, we ended up with 3/4 are purple, and then 1/4 of the plants were white. So the white begins to reappear. Why is that? So the all-purple F1 generation, or first filials, did not produce the light purple flowers as predicted by blending. So Mendel needed to explain why the white color seemed to disappear in the F1 generation, and why white color would reappear in 1/4 of the F2 offspring. So Mendel developed 4 hypotheses described below using some of our modern terminology. So the first thing, alleles are alternative versions of genes that account for variations in inherited characters. So alternative versions of genes, it's sort of like white and purple. Those are 2 different versions of that same gene for flower color. Okay. For each character, an organism inherits 2 alleles. Fun fact, if you're following along here, you get 2 alleles because they're diploid or diploid. So it has to be accounted for in each complete set of chromosomes. That's from that whole meiosis business. Okay? So we get 2 alleles, 1 from each parent. The alleles can be the same or different. If they are the same, they are called homozygous. Homo means same. I guess it would be better if I just underlined the same and put it there. Okay, so we have identical alleles. Both of them are the same. If we have hetero, hetero means different. See, better that time. And the different has 2 different alleles. Those are important terms to note. Although I can't speak, they're still important things to note. So if the allele of an inherited pair differ, they're different, then one determines the organism's appearance. It's called the dominant allele. Okay, the other has no noticeable effect on the organism's appearance is called a recessive. What I usually say is that the dominant allele sort of masks the expression of the other one. So it masks it. If we have different ones in a pair, then the dominant will mask the recessive. Okay, so the phenotype is kind of the appearance or the expression of a trait. I know it doesn't always work, but the pH kind of makes us think of like a physical characteristic. So sometimes that's how I'll describe it. It's sort of like a physical characteristic, that kind of appearance, physical appearance. The genotype, however, will be the genetic makeup of a trait. And we'll get more examples of those as we go along, but these are important definitions, incredibly important definitions as we move forward. Next thing, so a sperm and egg carry only 1 allele each, remember, because they're haploid. Since we just talked about those are haploid and carry half the genetic information of each of the parents. Okay, so egg and sperm, one allele each. Okay, so because the allele pairs separate or segregate from each other during the production of gametes, the statement is called law of segregation. And the fact that the parent originally had kind of two, I'm going to roll it out here. So the parent has two, actually, I got this better. Okay. We had two, oh my goodness, I can't even get it out. If we're diploid, we have the same things, but as we go through meiosis, we know that we're going to end up with 4 daughter cells that each have 1 in them. My God, it's the thing we practiced last time. So if this one has 2, each of these has 1, and it has half the genetic information. But it means that each of these that's available separates. Dear God, it is later at night than I would like. I'm not making excuses, I'm just saying it might be a reason for the issues of me speaking. So we'll get through it though. So Mendel's hypothesis also explains our 3-to-1 ratio in that F2 generation. So all the hybrids have what's known as a big P, little p genotype. Okay, so the big P would represent the purple color, and a little p would represent white. So notice the parent. So if we had, its parents were true breeding, and so it got a purple characteristic and a white characteristic from each one. So they would have big P and little p as the genotype. So when we do a Punnett square, we show all 4 possible combinations that those gametes could combine. So let's look at that. Okay, so our P or parental generations were both true breeding. So true breeding, oh my God. Okay, true breeding. I hope you can read that. That's terrible. I'm sorry for how bad that is. Anyways, true breeding means that they're both what we call homozygous. So these are homozygous dominant and these are homozygous recessive. So it's the same, but for the recessive. When you cross those, they're going to get 1 bit from each parent. So all of our hybrids are big P, little p. This is what that kind of cross would look like. So this would be 1 parent with big P's, 1 parent with little p's. And then all the little baby ones would have a big P, little p, which is what we see. All of them are big P, little p. You cross it this way and then this way, and that's how you get it for each of those squares as they align. Okay, so what we see with big P, little p is that when they actually go through the Punnett square, we separate big P and little p for each of them. And this is essentially segregation at this point. I'm having to write big because this one, segregation. It's the law of segregation. This is an angry stylist today. Okay, but what happens, so we cross the big P with the big P, and we have big P, big P. That's going to come out purple. Big P, little p. It still comes out purple because if the big P is there, it's masking the expression of the white. Then we get a big P, little p again by crossing those, and little p is here. There's no big P to mask that, so when we have the homozygous recessive, it will express as white, which is why in this particular generation we'll get back to the white flowers. All right. So a little tricky on that one, but we'll look at it one more time here. So a locus, good term here, plural is loci. It's the specific location of a gene along the chromosome. So for a pair of homologous chromosomes, like we talked about, alleles of a gene reside on the same locus. So at the same site, the same location, same home. All right. So homozygous individuals will have the same allele on both homologs. Heterozygous will have different alleles on each homolog. So this is what it would look like. So homozygous dominant, homozygous for the dominant allele will be big P, that's one here. Homozygous for the recessive, you can see that here. And these are homologous chromosomes with the same loci for those particular genes. However, on this last one, you can see it's heterozygous. It's different. We've got a big B and a little b. So this one would express as whatever the big B is, because it will mask the expression of the recessive allele. Okay? So that's another way to visualize that. All right, this one's a rough one. This one's a doozy, so this will be a good time to take a break. Look at your notes, make sure you sort of know what's going on, write a few questions, start that summary, come back. Okay, so we're not going to get into this too much. I'll explain it. Oh, I don't remember. Sometimes they do ask these on the STAR test, which I think is just a horrible idea, but we're going to work our way through it. So a dihybrid cross actually is mating of parental varieties in two characters. Two characters. So if we wanted to see what would happen, what's the probability of having certain flower colors and plant height. That would be what we're doing, is we're crossing for two particular characters. I'm sorry, I wrote all over that. Okay, so he performed the following dihybrid cross with the following results. Parental generation with round yellow seeds and wrinkled green seeds. In the F1, we saw all round and yellow. So that would tell us round and yellow seeds are the dominants. Then as the F2 generation, he found what's called a 9-to-3 to 3-to-1 ratio. Okay, here's the thing. 9 of them look like the round and yellow that we saw in a parent. 1 looks like the green wrinkled we saw of this parent. And then we've got an actual mix in between of wrinkled yellow and round green. The next one. Okay, so there is a tendency for us to look at gametes and do these wrong. It is a weird one that we'll actually, it's much better if you just practice this one. But I can show you a little bit about the idea is that you kind of have to cross them a little bit before you cross them. You have to get all the possibilities of gametes for each, which just happens to be big R, big Y, and little r, little y. Okay, and those would go all into the next generation. The issue then is that for these, it becomes a little weird. We can't do it this way. You've actually got to cross all these. So we've got possibility of big R with big Y, little r with big Y, big R with little y, and little r with little y. And those are both on each side. Again, this one's a gnarly one, but you can take a look at it. I'm happy to kind of revisit this one in class. I think that would be a little bit wiser than trying to do this now. It's a doozy. It's a doozy even for me. I don't always enjoy them. Ugh, okay, but what I will say, because the basic premise on that one is that we've got, let me go back. We've got two like the parents, and then you've got this combination of those traits, of the wrinkled and the green and the round, whatever. You've got a combination of those. So what that actually tells us is the important part, is that we've got new non-parental combinations. So what it suggests is that inheritance of one characteristic has no effect on the inheritance of another. That's what it's trying to say. So it suggests that a dihybrid cross is the equivalent to two monohybrid crosses. And so we call that the law of independent assortment. And basically it just says that each individual character is not dependent on the other. Okay, so that's basically what that's saying. And that is a key concept. The law of segregation and the law of independent assortment are two big ones for Mendel. So the following figure demonstrates the law of independent assortment as it applies to some Labradors, which are always so cute. But here's some black coat and normal vision. And then you can get black coat and blind, a chocolate coat and normal vision, chocolate coat and blind, and we'll still come up with that same 9-to-3 to 3-to-1 ratio if we mate double heterozygous black coat with normal vision. So it gives us some of that. I'm just showing it to you quickly. We can, like I said, we'll reevaluate. Now here's another key one, is that there is a test cross. So it's mating between an individual of an unknown genotype with a homozygous recessive. So a test cross can show whether an unknown genotype includes a recessive allele. Mendel used test cross to verify as true breeding plants. So we can look at the next figure to demonstrate that. So let's say we have this cute little black puppy. Oh, he's so cute. And we don't actually know what his genotype is, because if he's black, that means there's two possibilities. He could be big B, or he could be big B, little b. Oh my God, okay, little b. That's the possibilities. So you can't really figure this out any other way than something you absolutely know for sure. And we know for sure that if we have a brown puppy, look how cute this brown puppy is. If we have him, we know that he's going to have a little b, little b. That's the genetics. So we can cross the two of these. I guess they both can be males. So this will be the lady. And just cause. All right, we're trying to determine the gender of the lady. So we can breed the two. And what would happen is that if her genotype is big B, then all the puppies these guys have together, these doggies have together, would be all black babies. If, however, she has big B, little b as her genotype, then half the babies will be black and half the babies will be chocolate colored. Okay, so that gives us that genetics of that. But I bet you 100% of them would be cute. I should not be allowed. This is why I can't go to Petco when they have pet adoptions. Seriously. So using a strong background in mathematics, because Mendel was a math dude, he knew that the rules of mathematical probability affected segregation of allele pairs during gamete formation and reforming of pairs during fertilization. So probability scales range from 0 to 1. An event that is certain to occur has a probability of 1, and one that has no probability of occurring is a 0. All right. So there's some things. Oh, these are great. Okay, so the probability of a specific event is the number of ways that the event or outcome can occur. So to determine the probability of two independent events, use the rule of multiplication, in which the probability is the product of the two probabilities of each event. Okay. The probability that an event can occur in two or more alternative ways is the sum rule. So this one's kind of, if this will occur and this will occur, then we have multiplication. If we have the probability that it's one or the other, so if you have sort of an or statement, then it's the rule of addition. That's basically all that boils down to. If there's an and statement, it's multiplication. Or statement is rule of addition. So what that would look like is if we had that dihybrid cross and I needed to say, like, let's see if I can do this real quick. Well, let's use the little Labradors. And whether they black color and then, I don't know, eyesight. So I'll do that. But if we have this, we could go through that whole monster dihybrid cross. Or the easy way is that if I want to say, how many offspring will be, I want to know how many babies are going to look like this. So what you do is you first would do the cross for the bees. Okay, so the brown color, or the black color. So we do big B, little b here, and big B, little b here. And doop, doop. Oh wait, crap, I messed that one up, I'm sorry. Okay, so little b, I get 1/4 of the time. Okay, so then if I do, boop, I'm going to do it one more time real quick, I promise. E, big E, little e. If we do the whole thing, okay, E and little e, little e. Of this one, I have 2 of these, so 1/2. So that would say the probability of me getting this and this, I would multiply. So that says 1/8 of the whole time I will get that particular genotype. 1/8 of the offspring should look like that. So that's the multiplication rule. Addition rule is like if, so here's a fun one. If I need, if there's 1 out of a 2 chance that I will get heads when I flip a coin, And I want to also include getting tails. Well, what's the probability of having heads and tails be a possibility? We add them together 100% of the time. It is certain you will get heads or tails. Oh, I'm sorry, I messed that up. I want to get this and this, or if I get heads or tails. I told you it's a little later at night than I'd like to. So this is an and statement. Back it up, really back up. If I get this and this, then I multiply. If I say the probability of me getting heads or tails, then I add it. What's the probability of getting heads or tails? It's 1. It is certain. If I flip it, I will get 1. Okay, here's some of the stuff that we're going to talk about with pedigrees. There's some really cool stuff, but I'm going to leave it on there as reference for you, so it's in the notes section. But we're going to come to that one together, so I don't want to overwhelm us with the amount of notes. So let's start with that. If you get to a point and you want to look through for some definitions, you've got some time, that would be helpful, but we can go through that one in class together. So this is a good place to stop. Take a break. Look over your notes, all that good stuff, and then if you have any questions or don't understand something, shoot me an email.